Hello, everyone, and welcome to Probing Cell Surface Target Internalization in Cancer and Immunology. I'm Matt Thorne of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. We do encourage you to participate today by submitting any questions you have during the presentation. To do so, you can simply type those into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You can also submit any technical issues there if you're having any trouble hearing or seeing the presentation. I'd now like to welcome our speaker, Dan Beacham, Senior Staff Scientist, R&D, Protein and Cell Analysis at Thermo Fisher Scientific. For a complete biography on our speaker, you can click on the Biography window on the left side of your screen. And Dan, you can now begin your presentation.
Thanks, Matt. And a special thanks to anyone who's able to tune in this morning or afternoon, evening, where you are. And no, it's a thrill to join you guys here, and thanks to LabRoots for giving us this opportunity to talk a little more about this great set of tools we've been developing for the drug discovery of immunotherapy. And so I'll kind of just jump right in here. I've got a fair amount of technical content for our deck today, and this is something you can always kind of reach out to us offline for as well. We have Salesforce and so forth that can actually reach right into us in R&D. But here today I'm going to talk about, as promised, some pathways that we like to interrogate for cell surface target internalization and trafficking, and means to visualize those pathways as we've developed in the imaging and fluorescent microscopy space. And again, quantifying those processes. And then at the end, I've got a new section that we're pretty excited about, with a new sensor for endpoints in the lysosome pathway, in particular a new sensor dye that activates selectively down there. And I've also put in some examples of endpoint and live cell readouts of cell health, and even a couple 3D cultures. And the live cell piece is actually going to be key throughout here, because many of these assays do rely on intact cells or their enzymes and pathways to visualize. And so, I'm previewing here. There, sorry about that. Okay, so targeted cancer therapies come in a few flavors now, since they're sort of origination in the late 90s, and we see antibodies being used to either draw macrophagic or targeted killing cell types to cancer cells or their cell surfaces. But more recently, along with those new cell modifying therapies, we're now finding ways to selectively internalize and direct cell surface targets to a catabolic pathway, rather than just simply block the epitope as the early HER2 antibody-drug conjugates did. And so, the same technologies we use to modify antibodies with drugs are also, of course, can be used to modify antibodies with dyes and labels to visualize their trafficking. And so traditionally, people have taken advantage of free lysine residues sprinkled across the heavy and light chains of an IgG. And this makes for a convenient way to use our succinimidyl ester derived labeling reagents to attach fluorescent dyes in a single step to any tertiary free amine on an IgG or other protein. And it's fast, it's effective, it is at random, and so you have to be careful of to control the degree of label and some other considerations, like where you're labeling. You want to be sure you can avoid modifying too badly the target recognition sequence, for instance. But it's a convenient and easy way to simply label up an antibody and visualize it in either live or fixed cell endpoints. And oops, sorry, once again. Of course, there's a non-covalent method that we have developed here using fab fragments that are pre-labeled, and then these fab fragments are aimed to recognize the particular cognate species of your primary antibody. So if you have a human anti-EGF receptor therapeutic drug antibody, these are anti-human fab fragments labeled again with fluorescent labels that can very quickly just bind and then traffic along with an antibody target. And lastly, we have more site-specific methods. One actually precedes what I'm about to tell you here, and that's just good old cysteine labeling. There's a pair of cysteines here that separate the two heavy chains, and those can be utilized to label with reactive maleimide dyes. But the site-specific incorporation we've developed here is using beta-galactosidase to modify specific sugar residues that are only present in a pair on a heavy chain in a stereotype location, and these are modified to receive these clickable handles, azides that can be rapidly and very specifically conjugated to alkyne dyes. And this might be desirable for those wishing to have, very reproducible, careful placement and knowledge of the location of their label, or for that matter, payload onto an antibody. And of course, minimize any disruption with the antigen recognition. And so, the studies I'm talking about here all originate on the membrane and involve the internalization via clathrin-mediated pits and trafficking through a canonical endosomal maturation pathway marked in early stages by Rab5, and these compartments are matured and the appearance of Rab7 and ultimately LAMP, the lysosomal associated membrane protein. And this is typically used as an endpoint, but there's also a recycling pathway where cargoes have a choice to either return to the cell surface or enter this catabolic pathway. And in truth, we can very well enable this internalization and maturation pathway. The recycling pathway is harder, it turns out, but we have some good tools there. And I show one example in a slide later on. But the interrogation of the efficiency of sorting between degradative lysosomal pathways and recycling pathways is also of great interest. And to visualize these things, you really need to be in live cells, and that's where I'm going to kind of aim here with the next little message. Oops, where are my words there? Okay, so yeah, so on the left here, this is kind of this is our look-see platform, and kind of along the way, I'm just going to show you how how I'm able to do the quick check and then a quantification on the right with the high content microscopy. But in both cases, if you want to catch these pathways in action, cells need to be kept alive, and that's actually achieved with these on-stage incubator devices that attach to either the inverted microscope on my EVOS or install into the high content scope over here on the right. And the critical thing there is being able to sort of visually confirm that I can see a signal, for instance here, lysosomes, and over here on the right, then I can quantify it by using spot count or other intensity measurement algorithms to quantify the cell level and generate quantitative data. So I'll show examples of both, but virtually all the measurements, I think I got a couple examples at the end that are not, but virtually all these are going to be in live cells, and that's kind of key for these pathways. And so yeah, starting with the cell surface, I had mentioned before, we have a very easy collection of tools, either using reactive dyes, IgG fab fragments, and or direct conjugated platform. And as xenons, really, that's just the name of the product line we give to our fab fragments. And so we keep really careful control of the fab fragments, and so there's a reproducibility factor. Of course, they can be labeled with any of our dyes and any of our and aimed at any primary antibody. And then, of course, all the improvements in dyes we've made of recent with the Alexaflor Plus and so forth and many of the sensor dyes I'm going to refer to now are we've been developing for use in this fab fragment at utility. And so the nice thing you get with this is not having to react and purify your primary antibody. You can actually just mix in a lower molar ratio of fab fragment than antibody, and then you'll be guaranteed no free fab fragment, and can visualize either on the cell surface, as we show here with our traditional Alexaflor 488, 594, and 647 dyes. These are nice ways to. This is another comparison that people often want to make in this space: is cell surface versus internalized target binding. Oops. And they're, the nice thing about, as I mentioned, without having to do purification steps, the xenon fab fragments are great for these, sort of high throughput workflows. And so, my colleague Ryan here has just done this in a 96 well plate, but he's been able to use it to pick out, 3 active antibodies out of the another 93 that are done in triplicate, it looks like. So out of the other 1/3 of that. And having a very large signal to noise, these dyes are great for picking out. They give great Z primes in these HTS kind of platforms. Here quantified again in our CX7 high content instrument. The flow cytometry is another endpoint I won't speak to here, but the dyes are of course amenable to that as well. And in fact, I think the earliest xenon reagents were developed for flow, and we've now adapted them for use in these cell surface target internalization and trafficking pathways. And so internalization is really the business end of the effort in a lot of immunotherapies. And one of the other approaches we take here is, how do we know something is actually in a lysosome or an early or a late endosome? And I mentioned those marker proteins earlier, right? So the Rab5, 7, and LAMP proteins, these can all be labeled with a green or red fluorescent protein, and then visualized in your cell line of interest. And so we do that with an interesting construct here. And this is a modified form of the baculovirus that we call BACMAM. And this is it transduces mammalian cells by virtue of pseudotyping on the particle surface. And then, it carries our payload, which is astounding, actually can carry up to 30 kb. And the magic here is that by virtue of a CMV promoter, you get selective expression of your gene payload, but the polyhedron promoters of the virus itself are ignored by mammalian cells. So you have this nice purely sort of episomal and transient transduction mechanism, and that we use this to mark by fusion to LAMP, Rab7 or RAMP5, these compartments in the cells. And so I'll show some examples of that. And so you don't have to build the virus; it just comes in the mail, and it's a P1 containment you can just use on the bench top, and then transduce most cell types. I will point out this is a big hematopoietic cell audience. Unfortunately, this technology does not work with hematopoietic derived cells, but has pretty good tropism across many other cell types, adherent and otherwise. Primary cells also tolerate it very, very well, much more so than we see with lenti and AAV. So transducing, in this case, with say GFP or RFP forms of the Rab7 or 5, or in the middle we've got LAMP, you can visualize these compartments quite readily inside the cell. And so this will be a positive control for us a little bit later in visualizing trafficking of a cell surface cargo through these places. We also have these other more, I showed you a fluorescent protein solution here. We also have our dyes. These are dyes, the lyso tracker dyes first. Lyso sensor I'll refer to later, but lyso tracker dyes are small molecule dyes that concentrate in the most acidic compartments of cells, and that's the other feature of this pathway that I'm really going to speak a lot to. So these, as these endosomes are matured, I'll have a figure on this later, but we'll see, acidification down to the pH 4.5 level, and so these dyes, actually the lyso tracker dyes in particular, their superpower is concentrating and fluorescing in these acidic compartments. I'll point out that it's not the acidification that causes the fluorescence, it's the concentration of the dye. And that's going to be an important difference later on. But these make for pretty reliable lysosomal markers, also with some limitations that we don't have a fixable version of them, for instance. But more on that later. So in order to visualize the rate of cell surface target either cycling into the pathway or recycling back out of the pathway, and then the rate of movement between these compartments, we're going to leverage an important parameter here, and this is really key to the live cell. I mean, not to mention cells have to be alive to internalize and traffic these compartments. It's a very dynamic process. On the order of minutes, tens of minutes to hours. And so this is the other piece, and that's really why we've kind of used these on-stage incubator devices, because you either miss things early on, or you have to wait for an overnight event to get full internalization and trafficking. So being able to visualize constantly and know where things are is winds up being pretty key with understanding the rates that a different antibody may internalize the same receptor, for instance. And so again, these are measurements I'm going to take both in the left space on my EVOS, where I can just peek and make sure that the experiment worked. Take some nice sort of, I don't have, take some nice pictures, and then over here on the right, I can then drop the microplate into my high content platform and quantify the signals. And so what I'm leveraging here, as I mentioned, is the pH, the acidification of these compartments with maturation. So, and there are several processes that people associate with internalization, all at least in those involving membrane pinching and formation of endosomes. So the classical and most cited use of pH dyes is visualizing the entry of labeled bacteria by macrophages in the traditional sort of phagocytosis measurement, where we have dedicated cells that recognize these cargoes, and then they fluoresce in a time and use dependent way as the compartment gets acidified. Another example of a reagent set that we've built out here is tools to passively measure just the tonic pinocytosis or membrane uptake by virtue of these pH-sensitive dyes conjugated to a water-soluble dextran. And these also we'll see turn on, going from dim to brighter and brighter as they're acidified. And then these last two on the right are what we're going to be sort of most focused on here, is either cell surface target binding by ligand and its triggers of internalization, or as I show over here, antibody internalization. And so we have a panel of dyes to do this, and I just want to call out a couple of highlights of them. First, these are dyes that are quite unique to this suit, to this purpose. Traditionally, before the Frodo dyes were invented, people relied on the quenching of fluorescein to visualize acidic processes, and so watching a signal disappear is always unfavorable compared to the apparition of a signal in a dependent manner to the analyte. And so we developed the Frodo dyes, as in pH-sensitive rhodamine, if you will, to be able to monitor pH and to best enable the discrimination of of the outside of a cell from an early or late endosome or phagosome. And what I'll point out here is that even at neutral pH, 7 or 7.5, both of these dyes have a non-zero signal. These are dim dyes that get significantly brighter with acidification, and that makes them useful for visualizing the early stages in this maturation and trafficking. Here on the right is a new form of the dye, and this one we tuned it out into the Sci5 channel to make it spectrally distinct from the others, but it also has a functional distinction, and that is its pKa. So if you look across the bottom here, these red and green sensors have a pKa right at neutral. we tuned the deep red sensor down to 5 so that we would selectively visualize those cargoes only in the late endosomes and lysosomes. Um, and we'll see why that's important a bit later. But so this slide is really just kind of a demonstration of the first two of those pathways in the cartoon I showed. So macrophages engulfing labeled E. coli. What you don't see outside the cell is all the bacteria before they fluoresce, but as they are internalized, they generate bright signal. This can also be used in a solution phase, of course. The low density lipoprotein delivering cholesterol to cells through the native or scavenger LDL receptors can be labeled with photo red or green or deep red dyes and visualized in real time on, in this case, hepatocyte cell type. And so, understanding where the cargo ended up is something we're always interested in. And so I've got a few kind of a hodgepodge of some of the reagents I just referred to a minute ago. So I referred to the red version of in this case, a HER2 antibody-drug conjugate. You can see that inside the lysosomes, which are marked in their membranes are green, as marked by the cell light, the BACMAM GFP lysosome marker fused to LAMP. And then a couple of other dyes just for landmarking. We've got these nuclear dye. In this case, just a dropper bottle. If you see that ready probe, branding, that just means it's a dropper bottle formulation. But in 96 and 3-4 well plates, the version of this is just a Herxt dye. Can provide any detail you like on that. And then the plasma membrane marked with one of our masking dyes, and that helps with the segmentation offline in high content later on. So I'm going to just show a couple more examples now from that new long wave Sci5 pH dye, Frodo Deep Red. And as I mentioned before, phagocytosis examples here, just at these are actually relatively fast events compared to the uptake and trafficking of some of the antibody-drug conjugates we've modeled here. But this is on the order of an hour, that tens of minutes that the signal is appearing, reflecting the rate of the accumulation in those acidic endpoints of the phagosome. We also have we've generated labeling forms of it for DIY labeling of antibodies or cells themselves to visualize their internalization. And in here, this is kind of neat. In panel A, you can see I've marked early endosomes in green and the deep red dye in purple. And so the deep red dye is largely excluded or invisible in those early endosomes. It's probably there, but you're only visualizing the amount that has made it to the late endosome and lysosome, which is overlapped here with with our lysosome RFP. I've pseudo-colored it white so that you don't have sort of bad display, but the point being is that this co-localizes with late endosome and lysosome markers in every visualization. Again, cells have to be live because these pH gradients disappear if you fix the cells. So kind of a side note, these pH dyes, Frodo dyes, have also been made into cytosolic forms to visualize aspects of cell metabolism or cell health. And I'll just show this an easy example here, the dye on board and cells treated with a mitochondrial protonophore to release protons and generate a drug and a dose-dependent signal. And this is sometimes used as an endpoint in looking at cell health and the consequences of payload delivery. But there will be more on that later. So, bringing the focus through these other means of internalization to targeted and antibody receptor internalization pathways. Here, kind of switching to the new generation of these dyes first that have been really built just to address antibody labeling. And these are, if you see some branding there that says IFL, these are the sort of improved solubility and labeling efficiency versions of the early red and green dyes, and these are have been developed in some kits I'll show a bit later for for DIY labeling at free amines on your primary antibodies. And this shows that, the early generation one actually, it doesn't work as bad as we show here. It actually works quite well, but it did have you got poorer yields. It was some poor solubility in the aqueous phase, and so we like the IFL versions a lot better as antibody labeling tools, quite specifically. And so and in the same spirit, the deep red version is also an antibody labeling kit. And another example here of showing how the dye is absent in early endosomes, and I think actually this is the same slide, and shown as present in late endosomes and lysosomes in live cells, of course. And this quantitation down on the bottom comes by way of either a cuvette or high content measurement. And so bottom left here, you can see the Frodo red, sort of the legacy traditional dye. At neutral pH, as I promised, it has a non-zero signal. It's not an invisible dye, but it's a non-zero dim signal that gets brighter with a half maximal. Is actually quite fully activated in the pH of a late endosome, and actually will be quite bright, showing up in 2- and 3-fold delta in signal just from the early endosome into the appearance in Rab5. Outside of the cell at pH 7.4, 7.5, there's still quite a bit of signal. Here we've quantified the signal of antibodies labeled with the deep red form in the middle. And so at pH 7.5, you can see the dye is essentially off, and much more like a luminescence measurement, you have a signal that appears out of the darkness and giving very large signal to noise ratios shown in that third bar graph to the right. And so and the secret of that is actually in the molecular structure of the dye. If you have any interest in understanding more about the dyes themselves, again, you can reach out to our sales or tech support staff, and they'll either tell you themselves or put you in touch with us. But this dye actually opens and closes, but in a pH dependent manner. And so in the off state, it is off. Whereas these dyes over here actually just have a fluorogenic increase in signal with protonation, and so it's a non-zero signal that gets brighter, and that makes a big difference in detection, but also the specificity for those late endosomes and lysosomes really stands out here in the pH activation. And so in this case, on the bottom right, I've gone ahead and quantified the internalization of signal that I can see when I label up a HER2 antibody and visualize its internalization on SKBR3 cells. And the cute thing I did here is I wanted to make sure that the antibody was still specific for its target. And so from left to right, I titrated in increasing concentration of unlabeled trastuzumab so that I could, at the top end, at the bottom end, compete for and effectively block all binding and internalization of my label, and then show that it is competed away in a nice, clean, dose-dependent way. And in parallel, I referred to this a little bit earlier, I can visualize the transferrin receptor trafficking in and out of the recycling endosomal pathway, and I can show that my probe hasn't affected that at all. And so this is not something people often do, but it's something you can do, and I encourage it if you have doubts about the non-specific amine labeling of your therapeutic or other antibody in your probes, because many times people want to direct the antibody to a different part of they want to direct the label to a different part of the antibody. A lot of data in this slide. Here's the sort of demonstration of that improved solubility I showed of the red and green dyes over their original forms. And this is really just highlighting, again, that the improved dye gives improved labeling. And these, as I think this is just some of the branding now for the kits we've made around them, just showing that it works at a very small scale, 1 mg, or I think we have even 100 microgram sample labels up to 1 mg with no difference in the signal to background in each of these. And so showing that it's a nice DIY system. And again, we've quantified this in our high content platform. Oh yeah, I'm sorry. And there's kind of a cool control here. The signal to noise that you see here is actually a cell type that is specific for the drug, SKBR3 cells, for a cell type that does not take up or express the receptor for the labeled trastuzumab. And so you can, we can generate signal to noise at the cell level, either one cell over the other. But there are other ways to generate signal to noise, of course, either by competing with the unlabeled antibody or just simply no primary control. And so these, as I mentioned, the Xenon platform of the Fab fragments can just be put together very quickly in a screening context, either into a high content or flow interrogation. This is probably its most common use. You don't have to worry too much about anything beyond, yes or no, it is taken up, and yes or no, it's inside and a little bit brighter. This is a non-covalent method, so the dye is covalently attached to the Fab fragment, but the Fab fragment is non-covalently associated with the antibody. But we pretty reliably, in fact, I have yet to see an example of a place where they part ways, and you can interrogate that yourself with another label just on the antibody itself. But they all make it so far as the lysosome together. And I mentioned the different IgG subclasses that the xenons can be aimed at, and so these are just aimed at a series of antibodies present on these cells, interrogated in this case with flow cytometry, just showing the sort of the peak signals coming out in the wavelengths of the label from the dye and xenon. And ADC screening, of course, is I think one of the most useful superpowers. I showed you that slide earlier of the 3 samples pulled out of a screen that Ryan did in triplicate there, and then it shows up quite readily over background, and we've got some nice work in process now with some collaborators looking at Z primes of this activity and so forth. And so, this is just how you use it, really. And this is, I recommend, as I mentioned, that one of the secrets to no wash is having subsaturating concentration of the label against the dye. So you need to know the concentration of your primary antibody, and you generally put it in at a lower molar ratio. But really, as with any of these products, the first time you try them out, I recommend on your cell type and your conditions to titrate them. And so changing the relative stoichiometry of the Fab fragment to the IgG shows, that there's a signal increase, but using too much causes a knockdown of the signal, because you actually have nearest neighbor quenching with some of these dyes. So you kind of want to be careful of the degree, what we call the degree of label, or the valency of the label on your target. As with some dyes, over labeling actually can knock the signal down, or even just generate off-target backgrounds by being taken up in non-target positive endosomes, just by being present in the solution, because these are all no wash. I guess I forgot to mention that, and that's a critical piece. But these are for the being able to visualize these and leverage the in this case the pH gradient, again, means live cells and no wash conditions. And so we've put together this sort of panel of screening kind of packaged and specific dyes for this use in human and mouse, and there's certainly more coming. So, all right, I'll spend the last section here on the endpoint of that pathway, using a brand new sensor dye, which is kind of exciting for us here, because using new chemistry, as I mentioned, I've been able to show how we can use pH to visualize this pathway, both recycling and lysosomal. But here in the next slide, I want to leverage a specific enzyme activity that's only in the lysosome, and that's cathepsin B. So this is another depiction of that pathway, cell surface targets trafficking into early and later endosomes, and then ultimately a lysosome, where the in addition to LAMP, the catabolic enzyme cathepsin B can cleave substrates at a specific linker, and in our case, free a quencher from a fluorophore and generate a fluorogenic signal. And so cathepsin B needs two things. It needs acidic pH, so we already know that that is there in the lysosome, and then of course active enzyme, which will recognize our target sequence and free up a bright and a fluorogenic signal that is quenched at rest outside the cell or in the early parts of the pathway lacking cathepsin. And so, this is a reactive dye, first of its kind, and it uses the same chemistries I described to you before for the pH or the alexifluor dyes. We've got targeted as well as amine reactive forms that you can use to label antibodies either at canonical sites or at random. Again, another example of a live cell no wash assay where you have to have the cells happy and alive. In this case, my onstage incubator overnight while you treat the cells, and then you interrogate at the end either with the with some simple imaging on the EVOS or much more quantitative on the high content Cell Insight CX7 platform here. And so first thing we wanted to prove to ourselves, I showed it a different way before, is that, did my label alter the antibody? And so I asked that before by showing a competition, a dose displacement study with unlabeled antibody. And I also hinted how you can, take an antibody specific for one cell type by virtue of its receptor, like the HER2 against trastuzumab on SKBR3s, and then interrogate a control cell type that lacks that receptor, like A431s, and then, or for that matter, find a different drug, cetuximab, which recognizes receptors on A431s that are absent on SKBR3s, or for that matter, use the labeled copy of the IgG subtype that these antibodies are derived from. Each of these can generate a background, which is an important aspect of any signal measurement. People love to, sorry, just extemporaneize a bit here, but yeah, people love to stand on their bright signal. But in my view, the background is the most important of these measurements. Signal is a self-fulfilling prophecy in imaging, and so having a clean background and specificity stands above all. And so we go to the trouble to show that a few times whenever we put a new tool like this out here. And so this is kind of a cute movie that, again, is just, it's made possible only in live cell, but it's a time lapse showing. This is actually an example of a very slow process, but it's nice. It shows us a few things. It shows us that our dye is not toxic. Cells are alive and happy after 2.5 days, and and then of course you see without any media removal, you see the appearance of the label and its release from the quencher inside the lysosome. They're presumptive lysosomes at this point, but I'll show you that data next. And so, this is an example using that same BACMAM expression system, in this case to label the membrane of lysosomes with GFP and LAMP1. So you can see that our lysolite dyes inside of LAMP1 positive compartments. And there's the other aspect of selectivity is against the cathepsin itself. We want to be sure that the substrate that we've made for this enzyme, again, which separates, which rather pulls together a quencher in proximity to a bright fluorescent dye, that keeps that dye quenched until it is recognized and cleaved by cathepsin B. And so the CA74 inhibitor here is something we use to titrate onto cells and quantify the uptake. These pretty pictures are actually on by way of a confocal microscope, just to kind of highlight the location and proximity of these endosomes to the nucleus. But you can show in a dose-dependent way that you can inhibit cathepsin and see inhibition of the signal. And there's a and so yeah, this is it's one of the selectivity criteria we have, and there's actually some more data on our website. Oops. Let me back up here. There's also some more data on our website showing these cathepsin inhibitor function in the cuvette, showing that we have good confidence in the inhibitor itself. So this makes another nice control for the activation of the dye by cathepsin. We could put together a couple examples in 3D models. So whenever possible, we'll grow cells into a spheroid and then can probe either with our nuclear dye or labeled antibody-drug, therapeutic antibody in parallel, or look at control IgG isotypes. And see here, we've got another form of it coming out. I think this now is also coming out. Is actually, this is an old slide. I'm sorry. This is now available. We've developed the Fab fragment and cyclic forms of this dye as well. Let's see. I think I have a little section here on cyclic. Yeah, I alluded to it earlier. This is a targeted method for attaching dyes or payloads to antibodies, and it again leverages the presence of a pair of galactoside of sugar residues on the heavy chains that can be modified by galactosidase to contain azides, very small reactive moieties that can then be conjugated to our alkyne or S-dibo fluorophores and have controlled degree of label and specific labeling. And so, I mentioned spheroids earlier. This is now the Frodo, the pH sensitive version of the dye on trastuzumab. And taking a closer look, you can then ask, okay, so here's the dye on a spheroid in the control condition. So it has gone in. But if we attach a payload along with the antibody to the cells, we see that the green signal has activated. And that's a second dye that I didn't really have time to go into here. But again, you can reach out anytime and I'll give all the detail you'd like on the caspase activation dyes. These are enzymes that direct apoptosis in cells. And so the triggering of apoptosis leads to the activation of caspase 3,7, for which in a similar way to the sensor for cathepsin, we've made a specific sensor for caspase 3,7 that has a recognition sequence that frees a dye from its recognition sequence that can then translocate to the nucleus and give a bright and fluorogenic signal without wash. And so you can see the consequences of payload delivery and its presence inside of the cells with an assay like this. Similarly, so I mentioned before we have a fluorogenic dye that gets brighter as you activate caspases. These are more endpoint cell health kind of considerations, post antibody or post payload cell stress pathways. And so the induction of apoptosis appearing from top to bottom in green. But you can also see the disappearance of a mitochondrial mitochondrial function dye, and that's in the orange or RFP channel here, and that's a dye. One of our mitotracker dyes that fluoresces in proportion to the mitochondrial membrane potential. So healthy cells with a nice steep mitochondrial membrane potential fluoresce very brightly, and then as cell health decreases by virtue again of a delivery of a payload or a toxin treatment, you see the loss of mitochondrial signal, which actually precedes the onset of the caspase signal. In most cell types, it's not really terribly well represented here, though. And last is just another example. This is the example I was referring to. This is a signal that precedes caspase by quite a bit in function anyway, and that's the generation of reactive oxygen species. And this is another midpoint or endpoint that people will interrogate with respect to a payload or toxicity kind of treatment. And You know, again, by virtue of the enzymes having to be intact, pathways active, these are measured in live cell mode on our EVOS platform with the, oops, I missed one slide, with the onstage incubator. So I'll pause there with one parting note. I alluded earlier to, you can reach out to us directly in R&D. Our sales force is able to reach into us. We also have a customer research team that is really interested in understanding the the pain points you encounter. These are very informal kind of interviews, really designed to help us aim our hardware and our reagents better to problems that we can help with. And so I will thank you all for your time, and jump to the last slide here, and take any questions as they've as they may have come in.
All right, excellent. So we do have a couple poll questions we're going to run through real quick. So if you do have any questions you haven't sent in yet, we will get to those in just one moment. So the first poll question we have is, you can type in your comments there. Did the content of the webinar meet your expectations? We'll give everybody a minute or so there to answer that. Give it another second or so, and then we'll move on. I'm going to get those questions organized as we go through these polls. Are there any other topics you would like to see in a future webinar? Then we've got, would you like to be contacted by a Thermo Fisher Scientific sales rep? And lastly, would you like a quote or demo on the EVOS M7000 imaging system from Thermo Fisher Scientific? Give that last one just another second or so. All right. So now we will start working through the Q&A. Let's see here. We can start with how many lysolite dye colors are available?
Oh, yeah, I was supposed to have a slide on that. We actually, I described all the research I showed there, and is actually now published and peer-reviewed with a deep red form of the dye in the Sci5 channel. We have just finished work and commercialized a green, a FTSE GFP channel form of that dye as well. There are two.
Let's go with, are there limitations of using lysolite dyes for lysomal localization studies?
Yeah. Well, so I showed it has a potent turn-on signal that comes by way of the cleavage of the cathepsin recognition sequence in the dye. that separates the quencher from the dye itself. And so that means the dye cannot turn back off. It's not reversible. That can be probably not terribly important. The lysosome is considered kind of an endpoint, but I will point out a feature of the Frodo dyes, the pH sensor dyes, is that they're quite reversible. So you can see the induction of a pH signal as the dye is acidified, and actually watch it decrease back to baseline or non-fluorescent at alkaline pH. And so it kind of depends on your needs for the limitations, but so far as it's concerned in the workflows we've tried, it doesn't interfere with its neighboring dye channels at all. Really, it's got a nice tight spectrum, and its limitation, I would say, is largely in the functional domain. It's a one-way signal that just shows the turn on, but there may be others I haven't thought of here. You cannot, the deep red form, of course, you can't use at the same time as the lyso tracker deep red. But the let's see here. That I think that would be about it. It works well with fluorescent proteins and other more traditional imaging considerations, as I showed with the nuclear and membrane dyes.
All right, thank you. Do you know if the light cell works with endothelial cells?
Oh yeah, the cell lights. Yes, they do. Again, it's from cell type to cell type. You'll see differences in tropism, and I'll recommend. So I have a whole separate bunch of material on the cell lights as one of the developers, and so what our recommendation though is always to titrate. In the particles per cell count with every new cell type you try in the reagent class here. These by and large are active on, so they have a defined particles per milliliter in the vial that you receive. And so we recommend, titrating onto cells at on the order of 2 to 100, all the way up to 100 particles per cell. Most cells work in the area of 5 to 20 or so. So, but that really is more to do with the tropism of the cell as well as the endosomal escape of the message in the BACMAM system.
All right. Another one here. I think it's saying, does the lyso tracker pH a lysosome stable after the formaldehyde fixation?
Yeah. I don't advertise it here because we do lose a fair amount of signal. The Frodo dye in particular, of course, because it's reversible, the pH gradient needs to be intact. So typically with fix or shortly after, all pH gradients have diffused throughout the cell, and so you'll lose a Frodo signal almost certainly. The lysosensor dye less so. It is not. It is definitely there's signal loss with the lysosensor, but we haven't generated enough data to really put a number on something we'd stand by just yet. It's best used in live cells, and you would definitely suffer a knockdown in detection of internalization and those lysosomes after fix.
All right. Another one here. Are cell health live and dead endpoints needed to study ADC uptake? Can they be done independently?
Yeah. Okay. So that's right. At the end there, I showed the reactive oxygen dyes, this Cas-based dye, the mitochondrial health dyes. Those are all standalone reagents in and of themselves. And so really to multiplex them with your pH or lysosomal sensor dye, you just need to be sure that there isn't spectral overlap. But so they can be done together, as I've shown a few times here, or they can be done alone.
All right. Another one we've got here is, what role do clarithin coated pits play in surface receptor internalization?
Oh, that's Okay, so this is the mechanism of internalization itself. And yeah, so what people in general, so the biology of endosome formation it begins with the formation of small rings of the protein clathrin and mediated by actin filaments in the um, and this is actually, there's just a lot of biology that could be described here, is the formation of the endosome. And so I was just simply referring to active endosomal processes triggered by a receptor binding or antibody activation event. Tonic internalization happens by distinct mechanism, the pinocytosis, or for that matter, a different kind of active internalization with phagocytosis on macrophages. But the internalization I was speaking to throughout this talk, the antibody receptor targeted internalization is a well, now classically described, membrane internalization and trafficking pathway that is part of the natural regulation of any receptor. Um, its ligand will often internalize and cause changes in the expression and response of neighboring receptors, in fact. But we're just leveraging the acidity of those pathways. And so clathrin is just part of that formation of the endosome um, in living cells.
All right. I've got time for one or two more. Uh, let's go. Why are live cell conditions so important for analysis of ADC uptake and trafficking?
Yeah. Okay. So I showed a few time courses of activity. I showed one, on the time on the order of minutes or tens of minutes, and then another on the order of days, right? And so, in particular, antibody degradation can take longer than its internalization. You may want to parse those, and the again, as both rely on the activity of live cells. You need those intact gradients. You need those those active pathways. You don't know when it's going to happen with every new antibody you're screening. So you've got a 384 well plate of novel primaries, and they some of them may do their job in the first 10 minutes, and others might take overnight. And so being able to monitor continuously can matter quite a bit.
All right, and looks like we'll end on this one as we're getting close to time. What are some relevant controls for the specificity of a particular ADC or labeled cargo for binding and internalization as its cognate receptor?
Yeah, boy, I hope I satisfied that question in the deck, because yeah, so the specificity is of great concern to us. And I show some examples, either using a drug to inhibit the cathepsin that gives us a window into the specificity of the enzyme for its substrate in that compartment. I showed how you can compete with binding with unlabeled antibody and displace the binding of your label and prove that it's still actively engaging, that it's rather it's no longer engaging the target. And in fact, I showed a dose dependent curve of that. And if you see more than one notch in the rising phase of that slope, you can also identify additional binding events that are being displaced there. And then the third kind of control I showed was cell type control. So picking a cell type that lacks the receptor you're probing and using the probe there, and showing that generates a functional background. That's also often done in flow. But we like it in the live cell imaging space because it allows me to collect a bunch of imagery on live cells and have them as regions of interest with a signal to noise that I can either make against their own background on the dish or against a background of another cell type, also with the same region of interest, but lacking my receptor of interest. So a few ways to be sure of the signal you're seeing, and get better selectivity or specificity in some cases. But there's always going to be we really want to be careful about generating false positives with a turn-on dye. And so that's why we've taken such care to really characterize these for specificity after labeling.
All right, excellent. Well, thank you very much again for your presentation and your time today. I would like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's webcast. Before we go, I'd like to thank the audience for joining us and for those questions that you sent in. Any questions that we didn't have time for today or that get submitted during the on-demand period will be submitted to the speakers and will be followed up with using the information that you registered with. This webcast will be able to be viewed on demand, and LabRoots will alert you via email when it's available for replay. So we do encourage you to share that email with your colleagues who might have missed the live event today. So until next time, thank you once again, and goodbye.
Thanks, Matt. Goodbye.